I'm out here in a wheat field in the Njoro area of Kenya, about 108 kilometers northwest of the Kenyan capital, Nairobi. This wheat field is in a research center that is run by the Kenya Agricultural and Livestock Research Organization. And standing next to me in this wheat field is Dr. Godwin Macharia. He's the center director here at this center in Njoro. Now, Dr. Macharia, if I could kindly ask you to introduce yourself as well.
Thank you so much. Indeed, you've just said my name. It's Dr. Godwin Macharia. I work in this center, the Food Crops Research Center of KALRO, Njoro, where we focus on wheat research. And I serve not only as the center director, but also as the lead wheat breeder for KALRO.
And as we're standing here in this wheat field, there's a tractor in the distance. I can see it tilling the land there, and we can hear it in the background. Perhaps if I can start off by asking you, Dr. Macharia, what are some of the current challenges facing agriculture production in Kenya?
Oh, sure. Thank you for that question. There are a myriad of challenges that are facing agriculture production in the country. You want to think about right from production to the logistics, to marketing, and so forth. In terms of production, for instance, climate change and associated issues, for example, erratic rain, rain that comes in excess, things to do with the new epidemics of diseases like we shall be able to demonstrate today, and that kind of things is a problem. If you consider inputs, for instance, seeds, improved seeds or what you call certified seeds, sometimes are not sufficient, and that's a challenge. When you think about, for instance, on the value chain marketing, even when the farmer is able to produce, marketing also becomes often a problem. Going back, even the side of production again, in the country, you have found that agricultural land is steadily declining or decreasing. Most of the productive land traditionally is just being put to other uses. For example, settlement, and that also becomes a challenge. When you consider a factor like even when the farmer has a harvest, there's what you call post-harvest losses, which is a big problem. So there are quite a number of problems, not just a few, that we can consider when we think about agriculture production and particularly wheat production.
Now, how does climate change specifically affect food production, particularly wheat production here in Kenya?
Climate change, just like I alluded before, comes in with a bit of issues. For instance, most of our agriculture, 90% or thereof, and when I say agriculture, we readily can even imply wheat, is rain-fed. That means we rely on rain. So climate change is manifest by factors like, reduced rainfall, sometimes excess rainfall, which is not favorable, or sometimes when delayed rainfall is not favorable for production. But apart from rainfall, climate change also comes in with and for wheat, in this case, we have found this very critical, diseases. For instance, though we know since the wheat was introduced in Kenya many years ago, almost 100 years ago, there's been the issue of rust diseases, which again we can look at today. But as much as we have been working very, very hard to manage those diseases through breeding for resistance, you have seen climate change has brought in new diseases. For the first time since 100 years, we are talking about Fusarium head scab. It's a disease that is largely, for the most part, known to be, associated with the temperate lands, or maybe Europe or maybe in the US. But now we are finding it right here in the equator, and that is also, we are saying, is because of climate change for the most part. There are also other issues. For example, I may want to mention quelea birds. It's a big problem with our farmers in Narok, our key county for wheat production. But you should know that this problem is also now being associated with a new county for wheat, Laikipia. It's really the dynamic around those quelea birds we want to associate it also with climate change. And so there are many other things we can mention that climate change has brought in itself, which lead to reduce production of wheat in the country.
Welcome back to Africa Science Focus, the science and development show from SciDevNet. I am Ogechi Kianyao. That was Godwin Macharia, the director of the Kenyan Agricultural and Livestock Research Organization, talking about some of the agricultural challenges in Kenya. Godwin's concerns are mirrored across sub-Saharan Africa, where food insecurity is a growing problem. According to the United Nations 2024 State of Food Security and Nutrition in the World report, Africa has the highest percentage of its population facing hunger, and that is a staggering 20.4%. And the situation is projected to worsen. By 2030, a shocking 53% of the global population facing hunger will be concentrated in Africa. There are several drivers of food insecurity, including economic downturns, conflict, and as Godwin mentioned, climate change. To address challenges associated with climate extremities, scientists are turning to innovative solutions. One promising technology is gene editing, which is being used to develop climate-resilient crops like wheat. But what exactly is gene editing? Our reporter, Michael Kaloki, finds out from Dr. Anna Backhaus, a pre-breeder for cereals at the International Center for Agricultural Research in the dry areas.
We could say gene editing is when scientists transfer some little piece of the genetic code from one plant to another, right? And normally this happens anyway when, like in humans, when you have one mama plant and one father plant and they cross, they will mix their genes, and then the children of this cross will have a mixture of both of their genomes, right? But the issue is when you do this maybe in plants, that you maybe cross a very, very good plant that is a very good crop with a crop that's actually not very good at all because it's very old and it falls over, but it has one specific region that gives it a really good resistance maybe to a disease. So you only want this little piece. So normally what we had to do is cross them and then look at all the little offspring which have 50% from one and 50% from the other, and then do lots and lots of work to only end up with the little piece from one of the parents. But with gene editing, we can kind of pick the little piece and edit it exactly as we need it, and then continue. And I think the important thing with gene editing, so you can imagine it like little scissors that make little edits in the genome. But I would say that it has been proven to be extremely safe so far, because the only edits we make are mutations or edits that would naturally or could naturally occur as well, right? So all the plants we have little edits in their genome, and every time there's a new plant made by crossing or everything, there's a little new edit. And these edits can occur anytime. So it's just that we now found a way to make an edit in a very specific location of a genome and a very specific edit, but it could have occurred naturally. And we also don't, in gene editing, you can't transfer a gene, let's say, from a jellyfish to a maize plant. You can only make little edits that would maybe switch from one maize to another maize, or from a cereal to another cereal. So all of these would be possible through classic breeding as well. And I think that's where, so we know that the edits you make are safe, and actually that the technology is safe. I think has been proven quite extensively so far through scientific research, but of course it's always good to do more trials and more experiments. My work particularly focuses on trying to improve cereal crops, so mostly wheat, which is one of the most important crops like grown globally, right? And barley, which is used also extensively, especially by smallhold farmers in North Africa. And our challenge is to keep trying to improve these crops so that they're more climate resilient, that they're more resilient to maybe specific stresses that they're facing, but that they're also more nutritious, and that they're better for end use. So kind of all things you could think of. And this is what normally breeders do to kind of try and improve the crop itself. So we have easier management and better use. And the difference to GMO is then that in GMO, you can transfer genes from one species to another. So that's really, I would say, the big difference. So with gene editing, you don't do that. You only edit the genome of the plant you're working with. And I would say a lot of the controversial parts, though, are not only the technology itself and if it's safe or not safe. There's much better experts to talk to than me. But the discussion that often comes out of it is, well, if you do this and you need to spend a lot of money to make these edits, or the GMOs especially, who owns it afterwards, right? And who has to pay for it? And this is something, of course, we don't want to happen at all with gene editing, that someone has the right to the seed or to something afterwards, right? The technology still needs to be freely available, and we need to be able to share seed with a farmer, and the farmer needs to be able to share the seed beyond and use it again. So we do not want to create a way that this technology can be patented and used to make money for companies, right? It's to benefit farmers. So I think this is something that can, of course, happen with any new technology. And that's where we know that actually just very good policies and regulations need to be built to avoid that, and also to control what kind of edits can be made. So we want edits that, for example, reduce the amount of chemicals. We don't want to make edits that maybe allow us to put more and worse chemicals. So all of these are really complex challenges that can mostly be addressed, though, through conversations with policymakers and developing. Each country has, of course, the freedom to develop their own policies. We just hope that there would be a global consensus on the use of all of these technologies eventually.
Now let's travel to Kenya, where gene editing is being tested on wheat at a demo farm. Godwin is there to show us around and give us some insight.
This project, in general, is seeking to accelerate breeding and delivery of improved rust-resistant varieties to the farmers' fields, because that's critical. In the earlier questions you asked about some of the challenges farmers are facing, susceptibility of the varieties, some of the varieties out in the farms is a critical issue. So this project that we are partnering with our partners seeks to accelerate the process of breeding and delivery of improved varieties by leveraging the latest, breeding tools or breeding, approaches like gene editing, as it were.
And if you could give me a bit of, if I could ask you perhaps expand a bit more on how this project is going so far for you here at KALRO.
We are in the second year of implementing this project, and I would say as KALRO, as a PI, the KALRO PI for this project, I'm really delighted. It's one of those projects we are implementing, but I think we are making good progress.
Just to add, PI means Principal Investigator.
Oh, absolutely. Sorry, I missed it. Principal Investigator for KALRO for this project. So I was going to say, yes, we are making steady and meaningful progress in terms of our objectives. First, we got the lines or the germplasm from John Innes last year. We did the first screening. We saw a bit of a challenge. Some of the lines were not heading. That means they were not producing the seeds. We considered that they could be photoperiod sensitive, but we have just done a repeat planting this season. That's what you're seeing today. And because we are able to, provide some artificial lighting before planting those seedlings, we saw that that really helped, because now we can see some heading and we can get some seeds from this, which is also good for us to observe. But the whole deal here is that we are able now we have collected 2 sets of data last year and this year on rust resistance, and we are seeing a bit of variation, which we are assessing. This will continue. We are getting the next set of material, planted in the field. Also do a bit of some greenhouse and inoculate to disease. Again, trying to evaluate and hopefully get some that are resistant, that can now be incorporated in the main breeding pipeline.
Now, you said we might be able to have a look at the rust in some of the wheat. Would it be possible to see that where we are? Are there any plants here that you may want us to have a look at?
Yeah, from where I'm standing, maybe I can show you that one.
Okay, you're just pointing to the left here. Yeah. And there's some plants here. And these ones are interesting. These do not have what you're saying, a head. So the others I can see have like a cone, if I could describe it as a small cone-like structure at the top of the wheat. And this is, I believe, where the wheat grains are, right, in this structure?
Yes, just what you've just called the head or the ear.
Yes. And
it would come with that kind of architecture. It could be cone-shaped. They have all kind of shapes. Yeah, so you're right. That's why you have the grain.
Okay.
So you just said earlier on that one of the lines we are standing here, or the material here standing right here, doesn't have that structure.
Yes, exactly.
Some of these lines we received from John Innes Center that gene edited seem to be photoperiod sensitive. That's what we say technically. They seem to like more light, more than the light we receive here. Maybe here we may receive about 12 hours of light or thereof. But this line seemed to be asking for more light, more. It is for, that's why I say it is for period sensitive. So it's a line I said in a different set that I showed you earlier on. We exposed those to more light, and so they were able to head. But this one was not exposed to more light, so it may not head. It's not adapting well.
How would you know if it has rust? What would tell you it has rust?
There are several rusts that we are trying to describe here. The key one that we screen for is stem rust.
Okay, you're just plucking one of the plants.
Yes, I'm trying to pluck one to see whether I can be able to show you.
Okay.
We are coming in tail end of the season, so it may not be able to show you very, very well. But indeed, if you look at this stem that I've just plucked out of this line that has no head, you could see structures like I would call them pustules. In our language, we call them pustules, like kind of rough sections of the stem.
I'm just feeling that.
You can see, you can feel.
Yeah, and they're black in color or brown in color. They look like patches. So, for example, this to me looks like, if I was to say, if you're to a long piece of grass. I know this is wheat, but to describe it that way, maybe a long stem of wheat, and then it has got these black spots, brown kind of splotches, if I could use that term to describe them. Would that be correct?
You are getting it very right. First, of course, wheat is in the family of grasses. So again, you're not wrong.
It has the resistance, Dr. Macharia, but it doesn't have the head.
Yeah, so in this case then, and again, that's a good observation, because after all, it's not resistance per se that is important. It is resistant and the yield is in the head. So why this is important for us is that as much as it's not provided us the yield, it can become a very important, we also use a technical language here, a word, parent. In breeding, the material used to make what we call crosses or to hybridize, to bring the genetics together, the genes together, we call them parents. So this could be a good parent when you are trying to integrate or introduce genes of resistance in an adapted, high-yielding material that is acceptable. So what would happen is that you pick this as one parent. It doesn't have the head true. It may not have the yield, as it were. And in fact, genetically it has a yield. It's only that it has not been exposed to what? The light regime, right?
So do you foresee this project will have a positive impact on those wheat farmers who are going through this problem of wheat rust? And what do you expect as well the project will have in terms of impact for other Kenyan wheat farmers?
Absolutely. I mean, like I mentioned earlier, you are implementing, and we have been implementing many, many projects in terms of, again, developing the right technologies. Let me use that, heavy word, but let me say the right varieties, just to because I'm a breeder. We have done it, but I see this as one of those. This project has been one of those that might be very impactful. We are pursuing gene-edited, approach or gene-edited material for varieties that we shall release. We have used all the conventional methods without of success, but we are not yet there. We release varieties using conventional methods. They will be out there for a short lifespan, maybe 4 years, because of a phenomena, or what we call the boom and bust phenomena, where we succeed in putting resistant genes together in the variety, but then the disease on the other side is able to come back. So material is released there, it's resistant. Within 4 years, the disease comes back more vigorous, is able to affect that particular variety, goes down. What with this gene edited, because it's more targeted, with the gene editing, you're even targeting up to the level of the gene itself. The mutations you do out there is up to the gene. We consider the material that will come out of this project are probably most likely to be more durably resistant. Maybe, as a hypothesis, maybe better than the conventional material.
And when you finally have that preferred variety that you would like to have through genome editing, how would you plan to roll it out to farmers?
Right from day one, for this project, one of the activities we're doing is to sensitize, our local partners, and of course the farmer is a key partner. So we have a number of scientists that are also trying to, especially the social scientists, that are trying to help on that end. Again, sensitizing our stakeholders.
This gene editing innovation is still in the field testing phase, but it builds on pioneering work by scientists at the John Innes Centre in the UK. Professor Cristobal Uauy is a project lead at the John Innes Centre, and he shares insight into the specific genetic edits for climate-resilient wheats.
Just like if you can imagine in humans, there are some traits that are defined by individual genes, like the color of our eyes and so on. And some of them are a little bit more complicated, like intelligence and so on. So in plants, it happens the same way. Certain traits can be encoded or can be written by one of these pages of DNA, but others require many different. So in the case of what we're doing now, we're taking the first steps in this, and the major traits we're targeting are two. One of them has to do with disease resistance, and this is focusing on rust, which are a very predominant disease in wheat, and that is very rampant there in Kenya, and also in Ethiopia and in Egypt and all parts of the world. And the other one has to do with the nutritional content to increase the iron content of the wheat grain. And in the case of the rust disease or the disease resistance, the aim there is to make the plant be able to defend itself better, so that when the rust or these pathogen, these pests arrive, the plant or the farmers do not need to apply chemical sprays, and they do not lose their yield. So the plant will itself be immune. So it's equivalent to when we've had a vaccine, right? When we've had a jab and we can defend ourselves. We do not get sick. We don't need to take extra medicine. The plant, the same thing. The plant doesn't require extra chemical sprays, which we know are not the best solution to this. And the plant is genetically resistant to the disease from the beginning. And that means that there's less loss for farmers, but also less contamination. And this is becoming more and more of an issue, because with changing climate, many of the diseases we're facing are getting more aggressive, and they're coming earlier and more predominant.
What have been the results so far in regards to genome editing in wheat, and how scalable is it?
So currently, what we have done, genome editing we start the genome editing in the lab, then that goes into the glasshouse, and then from there it goes into the field, and then the next step would go into farmer's field. Currently, with wheat at least, we're at the scale where we're at the farm, we're at the field at the moment, and the next day we'll go into farmer's fields. And it's a little bit about legislation. So in the UK, we can do these trials. In Kenya, we can also do trials, and that's part of the project, is to take some of the varieties that are currently grown in Kenya, do genome editing to make them increase their resistance for this rust pathogen of wheat. And by the time in the third year of the project, to be able to do trials in farms in Kenya to test the performance, because at the end of the day, we do need to do the trials to see if the plants will behave. This is, in a way, this is science. So we have to do the experiment to then test it. And then, of course, based on that, we evaluate, and we work with farmers there and with local agents to see if it's valuable to then continue it. So we're doing those trials now. It's still at the developmental stage, but we're very excited that the initial results here have been very encouraging. And then the next stage will be to do the trials in Kenya, in Egypt, and Pakistan, and to see how the plants behave to those three different environments, which, because they are different, it means that the pathogens are slightly different. And we want to see if the change that we've done to the plant will maintain its efficiency across those locations. And the scalability about it is very scalable, because once you know that change happens, you can then transfer those changes by just doing the traditional crossing that we've done. So just basically doing breeding as we've done it for the last, 200 years, or you can actually do the edit as well. So it is very scalable technology, because the important thing is that technology is within a seed. So the seed is the one that's protected. The seed is the one that has more nutrition. And the seed is ultimately one of the most scalable technologies in the world, because you can multiply it, share it, and suddenly now you've multiplied it and it's scaled very quickly. So that's why our efforts are to try to put technology in the seed rather than apply technology with chemicals or other agents that we want to, in a way, avoid their use as much of.
What have been the overall benefits and pitfalls of genome editing in crops? And also are genome-edited crops nutritious?
Yeah, so in terms of the benefits and pitfalls, it's like any technology. Any technology that we use, from our mobile phones to, there are some things, a mobile phone can be great. We can have the M-Pesa. It's great to do our banking and pay. But of course, someone can take advantage of you as well. So like any technology, there has to be what we call responsible innovation. So we have to take it one step at a time. And that's why we do our first trials in the glasshouse. Then we go into an experimental station with KALRO and colleagues to test it in the field. Then we go to farmers. And then so it's a stepwise process that we do to make sure that we can evaluate the crop in the best way possible. Importantly, the changes that we make are actually made by the plant, as I said. The only thing that we do with genome editing is that we make a cut in the DNA and the plant will repair itself. And just like in any process, sometimes the plant makes a mistake and will introduce a mutation. So there's a natural process of making a mutation, and this breakage of DNA happens all the time. So when they tell you, don't put yourself in the sun too much because you might have a change in, you have to put sun cream or something, that's because UV light is a mutagen. It will break our DNA and our bodies will repair. The plants have the same thing. UV light or things will break DNA and the plant has a mechanism to repair. But sometimes they make a mistake. What we're trying to do here is to make that error happen, that mutation happen in a very particular place in the DNA that we think would cause something that will be beneficial for the plant. And that's what we're doing currently. So in terms of safety, it's a natural process, but of course we need to be responsible in the way we innovate and take it one step at a time. But per se, there's not riskier than any of the other breeding technologies that we use. There's nothing to say that it would be less safe than them. In terms of the nutritious aspect, so again, the changes that we're making in the case of the rust should not have any unintended consequence in the nutrition of the crop. And on the other hand, the other project that we have is to actually try to increase the amount of iron in the grain. And that's an issue because, of course, we know that iron malnutrition is a big problem in many countries. And often our wheat flours are actually fortified. They actually put iron into the wheat flour. They put little pieces of iron that are microscopic. So you might not see it, but if you take a magnet, you can actually get the iron out of your wheat flour, at least in the UK and many other places around the world. So what we're trying to do here is trying to make the plant accumulate a little bit more iron in the grain itself. So when you eat it, you're actually eating a crop that is basically supercharged. So you get extra iron. And that's very important for population, for public health reasons that we want to do that, because iron is a big thing in terms of malnutrition and anemia across the world.
Effective collaboration is crucial for this research. The UK CGIAR and the Consortium of International Agricultural Research Centres are supporting this innovation in partnership with KABI. Dr. Richard Shaw, Director, UK CGIAR Centre for Collaboration in Science and Innovation, and Senior Regional Director of Europe and America at KABI, discusses the importance of this collaboration.
My work here is about getting collaboration between the UK and the CGIAR system. And that was set up about a year and a half ago because there was a recognition that the CG, which is a remarkable research collective, was unable to access some of the research that's potentially useful in the UK. And there was a disproportionate gap between the UK potential skills and the use in CG as measured by publications. So we set about bridging that gap and making sure that the valuable research that UK institutes are producing or able to produce or having a big enough impact in global agricultural practice. So the idea was to support global food security by bringing together those scientists to form impact-focused research collaborations, impact being the key word. The three key objectives of the centre were strengthening the relationships between UK science and institutions and the CGIAR, making sure that there's a strengthening of the impact that they have, especially locally, and making sure that we align with the CGIAR programs and transformation initiatives, so without creating unnecessary friction with a whole new bureaucracy. There's a recognition that a good partnership can produce results far better than the sum of their parts, and they can improve the quality and reach and sustainability of the science that they generate, and a critical source of learning. So individuals and organizations could be pushed to challenge assumptions and build on knowledge. And I think that's the core to the skills that we all have.
Dr. Backhaus explores how gene editing can improve food security in Africa, building on her work at the International Center for Agricultural Research in the Dry Areas, a CGIAR center.
Because I see food security as a huge challenge that can only be addressed via multiple and really interconnected solutions, right? So this can go all the way from the field to the food production chain to the end, to the use and waste reduction in the household itself. But one of the solutions is to create crops that are themselves more resilient, more productive, and more nutritious, so that you can reduce the impact you have on the environment in the field where you grow them, so you can improve farmers' lives and livelihoods because they have more nutritious food and also more to sell in the market. And this is, of course, just one avenue into the whole problem of food security, but it's a very enjoyable one for me because I get to use kind of science and new discoveries to try to push forwards. And actually, plant breeders have worked on this task for many years quite successfully, and the CGR has played a role here by breeding crops and varieties that are improved for smallhold farmers in the global south. And so for centuries, these breeders have reshuffled the genes inside the plants to create better combinations and thus better crops. But since the discovery of genes and also gene editing, we kind of have more tools and more precise tools in our box of abilities to do exactly the same thing. And so if we can find, for example, a gene that confirms resistance to a disease of plant disease, or that can increase the amount of micronutrient content, and we can now take this gene and then edit into any crop varieties that is already grown somewhere that's already adapted, and thus kind of speed up the whole process of sharing beneficial genes around the globe.
Dr. Backhaus, what are the long-term global implications if this genome editing technology is successful?
Yeah, that's a very big question. So let's start small. So I think if our field trials are successful and actually can prove, for example, that one of these varieties we created is really a game changer in, for example, having this great new disease resistance to stem rust, which is what we're currently testing with KALRO, and then that would open a kind of a new discussion around the regulation and use of this technology, which is something we can't underestimate, right? We still need to open actually the way we can use this technology and the acceptance. And it would ideally really pave a way to increase the acceptance, because you always need a good case study, right? You need the really scientific proof to say that there's a benefit in using these new varieties. And this is what our data should really add to the discussion. And then hopefully thus enable the technology and the varieties created with it to be freely available to everyone. So I think that's really important to stress, right? We don't want to have patents here involved. We want farmers to be able to use this technology or the product of the technology very freely. And if that's successful, then, well, we can have new gene editing projects. We can help other scientists to work further on it. And really the very long-term implications would be that we would have a really powerful tool to improve varieties more rapidly. So there's a lot of, so all these edits we can make with gene editing, they are also possible through classic breeding, mostly. But it saves us time and it's more precise. And I think we can all agree that time is of the essence in a century where climate change seems to unravel faster than we can even comprehend, where new diseases spring up in plants, just like they did for us with the pandemic, right? The same is true in the plant kingdom. You can suddenly have rapid and pandemic spreading and destroying farmers' fields. You can have heat waves of unseen precedents in countries and loss of water. So really being faster and adapting crops to a multitude of challenges at the same time will be a game changer.
And that's all from us at Africa Science Focus today. If you want to find out more, head to the SciDevNet website, and that's www.scidev.net. Today's show was produced by Alice Hurst, with editing by Titilape Fadari and Ogechi Keanyao. and reporting by Michael Kaloki. I'm Ogechi Keanya. Until next time, it's goodbye. This episode was supported by the UK CGIAR Centre. The UK CGIAR Centre, hosted at CAB International, the parent organization of CIDO.net, aims to support global food security by bringing together scientists from the UK and the CGIAR to form impact-focused research collaborations. This project is funded by the UK International Development.